I'm Michaela Benzeval, and I come from the Institute for Social and Economic Research at the University of Essex. And I think the reason I've been invited to give this talk is I'm Director of Understanding Society, which is one of the ESRC-funded infrastructure projects that combines social and biological data to enable people to do this kind of research. And my background is in research on health inequality, so obviously I'm also, from a personal research perspective, quite interested in this. So ESRC define biosocial research as being concerned with the dynamics of the interplay between biology and people's experiences and behaviours over the life course, and having a multidisciplinary perspective, taking kind of expertise from both the biological, medical and social sciences. And I think there's been quite a big drive to promote and support biosocial research in the UK over the last few years. Personally, biosocial is not a word I ever use because if you have a bit longer in the tooth than some of these reports, it actually comes from a kind of field of discipline that's about biological determinism. So I prefer to think of this as being about integrating the good value of working across social and biological perspectives, both in terms of data and ways of thinking. And I think Pam Hurd summed this up really nicely, where she said in the medical world, what people tend to do is have all this really rich medical information about people's health, and then they throw one socioeconomic variable into their models as a control. And in the social science world, we have really rich information about people's incomes and family lives and social networks, and then we add kind of one self-rated health variable to try and understand these things. Now, that's obviously a kind of forced dichotomy between the two things, but the value really is in bringing those two perspectives together so that we have rich information on people's social circumstances and experiences and behaviours, and rich information on people's lives, health rather. So what are the ways and value in combining social and biological perspectives? So first of all, and perhaps the kind of key one thinking about as I run a data set, is that we can collect biomarkers, and I'll explain a bit more about them in a minute, but they're objective early indicators of biological processes and physical functioning, so they allow us to kind of look at people's health perhaps before people realise they have disease, and they allow us to look at people's health in a much more refined way, different aspects of health, which means we can investigate things differently or better. By thinking about the biological pathways that connect people's social circumstances and their health, or their health and their social circumstances, we can begin to kind of understand biology and causation. Because if you think that being poor leads to ill health, at some point there has to be a biological change that leads to that illness. When you move to think about genetics rather than just sort of biomarkers, then there's kind of the whole nature-nurture debate. What is the genetic, biological underpinning of some of the social phenotypes that we have? And that's perhaps, again, another false dichotomy that's been around in the literature for a while. So now we're thinking much more about interactions, gene-environment interactions. So, does the amount you might be affected by air pollution in terms of your respiratory health, is that affected by your genes? Also now we're understanding that although your genes are fixed at birth, they can be altered in the way they influence your life by the environment. So something called epigenetics, and again I'll say a bit more about this in a minute, that means they get switched on and off, and that can have an environmental impact. And the reason for doing this is it A, gives us a better understanding of causation of some of these relationships, and B, it allows us to identify intervention points for when we might intervene to stop the kind of poor relationship between people's poor social factors and health. So when we started looking at this in Understanding Society, we did a review, not a systematic review, but we looked at 10 years of journals in these different disciplines to see how many papers there were looking at using biological information in the research. So in demography, we looked at 3 different journals, and obviously different journals have different frequencies of publishing, so the ends are not very helpful. But we found a range of papers where really what they were interested in was the idea that having more biological information meant instead of kind of focusing on thinking about death, you could go back in the disease process to think about kind of pre-death, frailty or losing physical functioning, specifically about different aspects of disease, and then back further again to look at risk factors. In geography, the literature tend to focus on kind of how neighbourhoods might affect health, and rather than having self-report health variables, they tended to pick up biological markers of stress or biological indicators of disease. In sociology, there wasn't much empirical research using biological data. There was a lot of ethical thinking about, should we be looking at genetics and social factors? How does people's biology affect their identity and things? And that came as a surprise to some sociologists who felt that as sociologists interested in disease, we should really be thinking about how different diseases interact with people's social life and how that might affect how people manage their illness. And so there's quite a surprise in the sociology literature that people weren't really kind of using these sorts of data. In economics, there's been quite a lot of interest, for example, in how testosterone was involved in the financial market collapse at a few years ago. But more generally, economists tend to be using genes and genetic information in instrumental variable analysis in order to look at causation. And I'll give you an example of that later. And then in the kind of more interdisciplinary journals, not surprisingly, it's about that interaction between social stress, biological stress, and health. So to start with, I just want to go through the 5 reasons I said, or types of research that people are doing in this area, and give you an example of each of them. So biomarkers are defined as objective measures that indicate a normal biological process, a pathogenetic process, or a response to a therapy. And they can be anything from people's height, through blood pressure, through to a kind of range of biomarkers taken from blood analyses. And I think an important point to make here is, although they're objective, there's still a whole set of issues around measurement. So how well that height is measured might depend on the difference in height between the people doing the measuring and being measured. How well the blood's taken might be dependent on the quality of or how long they're in the post for and what the temperature is. And so there's a whole range of things that, nevertheless, despite these being objective measures, might affect the quality. So they're not perfect, they're just different. And one of the things that's important to say is this isn't just about disease, this is about the whole physiological function through life. And so a lot of the sort of indicators we might think of grow during childhood into kind of early adulthood and then begin to decline. So what we're interested in is kind of how they peak, when they peak for different people, and what the decline is. So this might be your lung function. So as a man, your lung function's peaks when you're 21, and as a woman when you're about 18, and then it declines. So if this was lung function, that might be a smoker, because their decline is much steeper than other people. So going to the first question about kind of what's the difference really between what people tell us about their health and how we might measure their health. This is a really simple comparison between England and Wales. So in the top graph, you can see that the proportion of people reporting their health as good is pretty similar across the 2 countries. These bottom 2 graphs are based on a biomarkers for inflammation. And inflammation is quite a good predictor of the onset of later diseases. It's sort of one of the first things that changes when you're not well. And as you can see there, that the Welsh have much higher, which is poorer, levels of inflammation than the English. So then we might think about, well, why is that? Is there some sort of stoicism in Wales, so they have more ill health, but they don't report it? Is it about awareness of illness? So these biomarkers manifest in you feeling tired. Is that different amongst different people, the extent to which you might then include that in your self-report? Does that vary by age or gender? So there's a whole set of questions about why it is that biomarkers and self-report data might differ, which doesn't mean one or the other is right or wrong, but that we can kind of understand a bit more about people's health, what makes people go to the doctors and things like that, if we can understand this relationship between your biological health and the way you report it and experience it. Another reason that having biomarker and self-report data together, again, is this sort of understanding illness behaviour. So this graph shows On the left, the blue bars is a biomarker for whether or not you have diabetes. The next red graph bar is whether or not you know you have diabetes or you report you have diabetes. The green one is whether you're taking diabetic medication. And then the grey bar at the end is whether any of these things are true. And so if you look at the kind of crossover between those things, you can start to identify people who have particular issues. So, for example, at the bottom here, the blue blocks are those people who have diabetes, so they know they have diabetes, but their biomarker tells us they're not managing it very well. So they're people who you might want to think about intervening in a particular way. The red bar is the people who have diabetes but don't know about it, or don't report it and don't take medication. So there's kind of a range of ways that having the combination of self-report data and biomarker data can lead to different strategies for prevention. So thinking about how having biological and social data together allows us to think about pathways, in the research on the sort of social pathways to health, you'll see lots of diagrams like this. And they're basically trying to think about how you go from people's socioeconomic position, which in this diagram is at the top, through a range of pathways to health. So, for example, if you live in poverty, you might not be able to buy a good diet, and that might affect your health. You might live in a kind of area with high air pollution. You might find not having enough money to get by or being unemployed psychologically stressful. You might not be able to afford to go to the gym. So there's a whole set of pathways. But the point is, at some point, you have to have a biological change in your body in order for that to lead to you having ill health. And so we can start to look at whether there's a connection between people's social circumstances, the biological change in their bodies, and then onwards to health. And then, so that's really thinking about the link between social stress and biological stress. And what happens when you're socially stressed is there are these 3 levels of response. So the first one's a very kind of automatic one, when you get up to give a talk or when you start to run a race, your heart rate goes up, cortisol rushes around your body, and hopefully that enables you to perform better if that's what you're doing. But otherwise, if that's happening to you every day, because actually you're getting up thinking, how am I going to feed the kids today? Or how am I going to get a job? Then that starts to damage your organs, those things happening repeatedly. And that leads to this secondary response, which is once that initial adrenaline rush or whatever cortisol rush has happened, these other markers start to change. And so inflammation, the graph I showed you earlier, is one of the key things that happens quite quickly after the initial response. And so we can start to look at some of these things in terms of whether they're happening before it's actually turned into the third response, which is disease. So one of the things that enables us to think about is when, for example, inequalities in health emerge. So with this graph, the lower blue line is people with degrees, and the grey line is people with no qualifications. And so you can see this is CRP again, and the kind of highest level of CRP you would have is 3. So you can see that inequalities in this marker by education are emerging in people's 30s, and they're at their peak in people's 60s, and then they begin to close up again, although probably that's more about survival effect, that we're not interviewing the people, or obviously we're not interviewing people who've already died yet. But thinking about when the inequalities emerge kind of helps you to think about intervention points that you might want to address. So now I'm going to talk a little bit about the genetics side of things and how it is that genetics might help us in social science research. So you inherit your genetics from your mum and dad, half from your mum, half from your dad, and you have 23 chromosomes, which is that illustrated by that kind of purple X, and inside of them is this double helix, which is where all your DNA information is stored. And although your genes don't change, as I said earlier, and we'll say later, there's kind of ways that they can be switched on and off. But what we measure in genetics research is this single nucleotide polymorphism, which I'll never say again, SNP, which is where in a population level there are differences between these base pairs in people's genes. So in understanding for society, for example, we've measured 500,000 SNPs, and we're now done imputation to take that up to 10 million. So there are a lot of these differences in the population. and people use that as a marker to do genetic research. So thinking about that sort of thing, you may remember in the news a few years ago, Dominique Cummings talking about how education was mainly genetically determined, or educational outcomes were mainly genetically determined. And that came from a lot of research based on twin studies, which is how a lot of genetics research was initially done. But now that we're getting genetic data in more general population surveys and observational surveys, this research is all being looked at again. And so a recent review of over 100,000 people looking at this actually found that genetics only seemed to explain about 2% of the variance in educational attainment. So I think by combining social kind of designs of social surveys, observational studies, thinking about education in different ways and looking at genetics in that perspective, we'll be able to sort of think about the balance between genetics, nature and nurture in a different way. Then also, we need to think about the interaction between people's genes and their environment. And so again, after this initial work, which sort of was very didactic, nature or nurture, we're now beginning to think about this interaction. And so there have been a range of studies that have tried to say it's not just how you behave might be affected by your genetics in a certain situation. So, for example, whether or not physical activity helps to reduce your obesity might be influenced by your genes. How drunk you get when you drink might be influenced by your genes. And then from an epigenetic point of view, if you keep getting drunk, in this example, that kind of might affect how your genes are expressed in the future. So there's beginning to be some research that looks at this interactions, but a bit like the kind of straightforward genetic research, it's mainly so far been based on twin studies, and we're just beginning to think about how to move that into the general population. There are huge issues about statistical power. When you look at some of these genetic studies, they're talking about having hundreds and hundreds of thousands of people, and that means no one study can do it alone. You're having to build up consortia, and then you have a whole set of measurement issues about whether you're measuring the exposure in the same way in the different studies or the outcome, whether that each study has the right SNP that you want. The second way we're thinking about the interaction between gene and environment is your environment affecting your genetic expression, which is known as epigenetics. So although your genes are your genes are fixed, how they're expressed can be switched on and off, or it can be strengthened or diluted. And that's done by a process called DNA methylation, where sugar attaches itself to your DNA. And there are kind of global ways of doing that, or global ways that happens, and then it also happens for specific biological processes. And so there are beginning to be some studies looking at whether methylation is indeed associated with socioeconomic circumstances and health. And so their SOBID study in Glasgow, which is a study of deprived populations in Glasgow, has looked at global methylation and found an association both with socioeconomic factors and heart disease. And EPIC in Italy has found that there was a dose-response association for socioeconomic circumstances for methylation, specifically around inflammation. So we're at very early stages, really, of thinking about the kind of social science value of some of these things. The last way that social scientists are beginning to think about using genetics is by using genes as instruments. So in economics in particular, worrying about collinearity, there's a long tradition of using instrumental variables, but finding those instruments is quite hard. Whereas if you use a gene as your instrument, so say you're interested in this case, obesity and mental health, you use a gene for obesity, then that gets rid of some of those kind of confounding issues. But actually, when studies have been done using this, they've tended to use one SNP of the 10 million that you have in your body, and that's kind of not really very effective. So again, there's a whole set of new methods we need to think about in thinking about how to bring genetics and social science together effectively. And one of the key things that's happening at the moment is a number of kind of social science studies doing genetics are creating what's called polygenetic risk scores, which are bringing together all the genes that are associated with obesity to help you think about these issues more. So just to come on to somewhere you might go and do this kind of research, if that's what you're interested in, Understanding Society is a household panel study. It was started in 2009, and it had, at that time, everybody from 30,000 households was interviewed. It incorporates the British Household Panel Survey, which began in 1991, and has 8,000 people. And it's a multi-topic study, so it covers a whole range of social, economic, behavioural data, and we collect that annually from everybody in the family, or rather we collect it from those over 10, and we ask parents for those under 10. And in the second wave for Understanding Society and the third wave for the BHPS sample, we did do a nurse visit where we collected all these kinds of data. So we have physical measures, we took blood, and we now have a range of blood analytes. We've done a genome-wide scan, so we have the whole information on the whole genome available. And we're currently doing some methylation research, or methylation analysis on some of those people who are in BHPS from the start, so that we'll have 20 years of kind of social data on them, their epigenetics, and then subsequent information from the study as it goes forward. And ESRC have funded us to try and build capacity around doing this kind of research. So we're doing both research ourselves and a whole range of activities to try and enable others to do this kind of research. We're an interdisciplinary group. I'm waiting for my 2 minutes. And we've kind of got a whole range of activities that we're doing, so you might be interested in. One of the things that we're doing is about to advertise some fellowships. So if you are interested in doing research in this area, then please come and talk to me afterwards. Perhaps 2 things to draw your attention to. We have some international visitors coming to give masterclasses. So next March, Teresa Seaman, who's the person who is in the research group that created allostatic load, I'm sure that you've heard of, is a kind of marker for biological stress, is going to be here next spring doing a masterclass. And Dan Benjamin, who's an economist who set up the Social Science and Genetics Consortium, will be here in March 2018. And if you're interested in getting hold of the Understanding Society data, this is where you can get it from. Thank you.